For the first time, we are able, from the sequence alone, to actually predict and make custom models for a very large range of application. And specifically, what makes a lot of sense for our industrial partners is to be able to predict like protein levels, for example, or in many different tissue types and across many different crops, for some of which might not have the same amount of omics data gathered.
Hi everyone, welcome to Let's Talk Research, the official InstaDeep podcast where we dive into cutting-edge AI research and the stories behind it. My name is Bora Gilola, I'm a research scientist here at InstaDeep.
And I'm Massimo Bortone, also a research scientist at InstaDeep. Today we are picking up the discussion where we left it last month on the theme of genomics, but this time we're looking at applications and real-world use cases of how to apply our family of nucleotide transformer models to tackle real-world challenges.
Exactly, and joining us today is someone who works very much at the intersection of development and application and deployment, very own head of applied AI for genomics, Marie Lopez. Welcome, Marie.
Thank you. Thank you both for having me.
So Marie, before we dive into the applications, let's talk about you a bit. What is your background? How did you get into genomics at InstaDeep?
Yeah, so I think I first got into bioinformatics and genomics because I was a terrible experimentalist. It was much easier for me to work on a computer. And I was truly amazed by the DNA in itself, the fact that it's so universal and at the large scale, it's so adaptable also to face the different challenges that you might find in the environment. So during my PhD, I actually studied how the apparition of agriculture 10,000 years ago has shaped the architecture of human genomes in human populations. And what is the legacy that we have today in terms of number of deleterious variants that we carry in our genomes and how this is shaping complex diseases.
Well, so you actually looked at the discovery of agriculture and how that affected humans at the molecular level, you could say. Yeah,
that's correct. So by comparing the DNA and the genomes of rainforest hunter-gatherers in Africa, and agriculturalists, we were able to pinpoint what were the changes that were introduced by living in the same place, for example, instead of having like a nomadic lifestyle.
Wow.
Getting in touch with zoonoses, which are the diseases that you can catch from animals. And all of this has shaped differently the immunity, for example, but also the demographic histories of this population has shaped the number of mutations that can cause diseases in their genomes.
I feel like we could talk hours for this, but maybe we should move on.
Yeah. So maybe, long story short, I think that's a huge opportunity with AI now that we are able to catch the different patterns that we might see across the evolutionary tree by looking at the sequences themselves.
Yeah, I mean, I'm sure we'll come to this a little bit later as well, but last month when we were discussing the NT family of models with Bernardo, we talked a little bit about kind of the volume of data that they're trained on and kind of what the different applications downstream essentially capabilities are. I'd be really keen to hear kind of your take on what you think these models are capable of and how you're thinking at applying them in practice here at InstaDeep.
Mm-hmm. Yes, so in my opinion, when you're looking at the genomic sequence, of course, you have all of the elements that you need in order to make a prediction, but at the same time, the DNA is highly dynamic and highly probabilistic. So there is some kind of paradox where in order to make a good prediction, you need a very long context around the DNA, around the single nucleotide that you're looking at. But at the same time, you want the prediction to be extremely specific for this one letter among billions. Mm-hmm. So in this case, having some kind of verticality in the training is very helpful. So training on many different species, so the 150,000 species that are introduced on the NTv3 model, but also pairing that information with actual omics data coming from the lab that are going to tell you how are the sequences behaving in different contexts. So I think that the future for this kind of models is to actually unify all the information to be able to juggle between this very long context and this ultra-specific predictions.
Mm-hmm. This might be a stupid question, but How do you take into account evolution when you train on so many different species? Is that something you need to do, or?
It's intrinsically taken into account, just because you would, by having in your training data DNA that is coming from bacteria to human, having like chicken, having mice, elephant, you're actually learning something about the actual experiment that has been carried out by evolution throughout all of this history, because whatever is working is going to stay, and whatever is not leading to any meaningful adaptation is not going to be kept in the species and transmitted to the next generation. So in a sense, you're catching on this evolutionary experiment just by having a huge diversity of genomes in your training.
It's in a way similar to how the kind of state-of-the-art protein structure models are doing this explicitly, right? We provide a multiple sequence of alignment of related proteins, and so the evolutionary kind of history is, so to speak, traced through those. But you're actually learning those relationships and kind of what nucleotides correlate with what other nucleotides and regions and subsequences and so on and so forth from the get-go, right? You're doing that essentially explicitly. You're teaching the model how to do that.
So not explicitly in the sense that we're carrying some alignment experiments, for example, but by feeding this training data inside of the model, you would catch like some kind of the grammar of how a gene is constructed, for example, what makes a regulatory sequence efficient, or what makes it tissue specific, for example. So we were able to prove that this learning is happening in the model because of how easy it is to fine-tune and to transfer everything that has been learned to sequences for which you have much less information.
Yeah, exactly. So the model is extracting those evolutionary features itself. And so kind of you now have this very powerful set of capabilities, right? You have resolution at the single nucleotide level, but also kind of really, really wide context around the sequence that you're interested in, right? Maybe it's the gene that you're interested in, but you also have given the model the information of what surrounds it in terms of the actual DNA sequence. What comes after, right? Or what are the different applications? What are you particularly excited about, and kind of where do you see that fitting in?
Yes, so I would say it's highly dependent on the end goal, but for the first time, we are able from the sequence alone to actually predict and make custom models for a very large range of application. And specifically what makes a lot of sense for our industrial partners is to be able to predict like protein levels, for example, or in many different tissue types and across many different crops, for some of which might not have the same amount of omics data gathered. So in a nutshell, if you are able to predict like the protein abundance in tissue-specific way in maize, this is the first brick towards having some kind of in silico screening. It means that in terms of being cost efficient and time efficient, you're completely changing the scale at which you're working, because you're now able to screen millions of sequences in just a couple of minutes on a computer before even planting a single seed, let's say.
Oftentimes kind of in industrial settings, right, the problem that someone is working on is highly, highly specific, and they are often very interested in just that thing, and will have a lot of very specific data around that. So maybe various in-house assays, so on and so forth, that don't necessarily exist across all of the training examples that you might have to hand, right? So what's the kind of what are the capabilities here, right? So you have this base model, and then you have maybe customer specific or project specific data that you have access to. How do you marry those two things together?
Yeah, that's a very good point. So I would say that here being able to model with AI the different data types and data modality enables connecting the dots between them and missing the blank, filling the blanks with the transfer learning. So let me give you an example. Let's say that you have a lot of data for a specific assay or many different assays for a specific crop. you would be able to feed that inside of the model and to make now a customized model for your application and extract some of the value that was just sitting on your servers, just because you are now able to unify all of the different data types into a single model that is performing some kind of deep understanding of the patterns that could lead to better prediction.
And this can be done on a kind of per project basis if need be as well.
Yes, absolutely. So this is the way that we would be engaging with partners. They would have generated data, and we are able to use this data on top of the model that we already have to actually fine-tune it and make it like a super model for the applications. This is something that we've been evaluating with tomato, for example, that has been specifically held out from the training species. And we can see that even if the model knows nothing about tomato, it's able to make very good prediction just by being fine-tuned because it's using all of the pattern it's learned about DNA and how DNA works.
And so customers, how do they interact with these models? I guess if they are machine learning experts, it's fairly straightforward, I assume. But if they're not, if they're more on the applied side, what kind of ways do they have to interact with the model?
Yeah, that's a very good point. So I think that we are at a stage now, and specifically for AI in biology, where one of the questions is like, should we buy the solution or should we build the solution? And it's very different in terms of how much machine learning engineering you put into the development of those models. And the way that we interact with clients on it is through the deep chain platform.
Okay.
So this platform actually enables scientists to use their data and on a click and button fashion to fine-tune the models, the NTv3 model, to their specific application. So we have a lot of all of the machine learning engineering is taken care of by the platform directly, the parallelization and the compute usage as well. And we have been also optimizing the models to run like 12 times faster than previous models. So it makes it scalable and usable in an industrial setting.
I mean, this is super important, right? So I'm in a very similar boat to you in that I was also horrid at experiments, but worked very closely with experimentalists. And there was always this almost divide between the experimentalist and the computational side of things, because you kind of had to translate things or go and do something for an experimentalist because there just wasn't that easy access to interacting with whatever the tool is, right? This goes for traditional non-AI computational biology tools as well. And so it's super interesting to hear that you're kind of thinking very deeply about how do I actually allow someone to interact with these models.
Yeah, I think that we do need a lot of expertise from the bio side. We do need a lot of domain knowledge. And this should be enabled with none of the complication along the way regarding like compute efficiency or compute usage. So we want biologists to do what they do best, which is like thinking about the data, thinking about the biological problem, and to use the model to the best of their abilities, not having to, as you said, like running this fragmented tool and unify all of this under this deep chain platform.
And yeah, I mean, I couldn't agree more. It's much easier to bring the tool to the expert than bring the expert to the tool, I guess. But I'd be curious. So I know that the family of nucleotide transformer models is pretty big. And among those, we have different variants, and one of them actually made the cover of Nature Machine Intelligence, a model called ChatNT. Can you tell us about ChatNT a bit more and how it differs from the other models in the family?
Sure, yeah. So I think that ChatNT was a very interesting project, and it was the direct follow-up of using language models to understand DNA, and then can we use language models to actually talk to the DNA models and to frame that into a way more like human friendly way. And we know like from experiments that natural language processing that using English to actually query the DNA models can help generalize across the task, just because, as I was explaining before, in biology, all of the tasks are more or less related: DNA expression, protein expression. So we might benefit from having a single interface to actually interact with the model. So that being said, the ChatNT enables this and enables to actually talk directly to the model to actually have it perform the downstream application and downstream task. So that's definitely the next step towards having more generalization and more user-friendly interface around biology for genomics, AI for genomics. Sorry.
Yeah, I guess it's pretty hard to beat natural language in terms of interface. I mean, it's been stress tested throughout history.
I guess so.
Yeah,
That's
people
how,
are really comfortable with it. Yeah, very cool.
So, Maria, we've talked a lot about kind of how these models are really, really powerful, but also kind of easy to use both with natural language and also with a kind of bespoke platform that makes them available to customers and users. I'd like to talk a little bit more about kind of specific industrial partnerships that we have here, right? And maybe taking it a little bit back to your experience in your PhD as well, and kind of looking at agriculture specifically. So we have this partnership, obviously, with Syngenta Seeds that has been long-standing. Would you be able to kind of give a few examples and kind of discuss the work that you've done there and how that all ties together?
Mm-hmm. Yeah, exactly. So we have this multi-year partnership, so a strategic collaboration that we have established with Syngenta Seeds specifically. And I would say that amongst the big challenges that are now arising in precision agriculture is definitely the architecture of complex traits. So complex trait in the sense of you have some measurable phenotypes. For example, is this maize line going to give more or less cob? Is it going to be resistant to certain pathogens? Is it going to be resilient to changes in climate, for example? So all of this is of very high importance for the field of precision agriculture and for food security as a whole. So the work that we're developing in that space with our partners is actually to enable them to screen many different sequences before taking any decision on what to do with them next. So there are many ways that we can actually do this, but we have seen with the ability of the NTv3 to tackle many different tasks and also prediction on how the sequence is going to behave. This is extremely valuable to teams like Syngenta Seeds because they can actually run many experiments on the platform and they can make like informed decisions like never before in a sense.
And I imagine that kind of ML is really good specifically in this kind of field of agrobiology and kind of crop engineering, because I mean, correct me if I'm wrong here, right? But we kind of have a much deeper understanding mechanistically of how something like human biology works, just because there's been so much more research in it. And so when you're dealing with these species, essentially, where we have just less breadth in terms of what we know about it, and also you're dealing with a bunch of different species, right? Not just one. There's kind of this added benefit that you get from a black box, an ML black box that really understands kind of the data that goes into it, without you having to be able to kind of, as a biologist, sit down and really kind of at the molecular level being able to explain how a change in one DNA sequence results in some phenotype.
Yeah, that's a very good point. And actually plant genomes are a little bit crazy, I have to say. Yeah. You would find like polyploidy, which is like instead of having only 2 copies, you would have for sugarcane 12 copies of the same chromosome. So and genomes are extremely large. You have duplications everywhere. So it's as you said, it's difficult to study. It's difficult to gather data for them, and this is where we see that these models that learn from many other different genomes and many different species can actually bring something to the table. And this is true also for orphan crops. So for rice or maize, you would have like a lot of data and that have been generated. But what about other, like cassava, for which you might have like less data available? You now are bringing all of the other information that you've captured in the inside of the model, bringing to helping understand more of this like orphan crop genomes. And you've been touching something that's extremely important as well, that is the interpretability of the models. So ML model used to be like very black boxy, and you get only the prediction in the end. But there was a true effort in the development of NTv3 to actually make it interpretable and to kind of trace back what has the model learned in order to make the prediction. And we've been able to see that the model can distinguish from regulatory sequences having like the most impact, for example, and this being in very good a link with what you observe experimentally. So yeah, I think that these two, this is extremely important for industrial application, right? Because you wouldn't move forward with a prediction. At some point you need like the mechanistic understanding behind that.
Yeah, you need to explain why, right?
Yeah, exactly. Like biologists would definitely prefer to have like the mechanistic understanding behind.
And it's also super promising that kind of, even though there is all this kind of diversity that we have across plant genomes, there is still kind of generalization across the different species, right? Just by learning from lots of genomes that we have abundantly, you can still extract lots of meaningful signal on genomes that maybe are a little bit sparser in terms of the data that we have for them.
Yeah, absolutely. And you can actually, the more data you're going to give your model, the best it will be at specializing to the task and to the species. But you're already doing quite a decent job on a zero-shot prediction for a genome that hasn't been seen with the base model that we have. But of course, you would like the more data, the better the model. That's a very general rule.
Yeah, maybe to tie it back to your background, I'd be curious to know, I mean, we're now entering this age of, I guess you called it precision agriculture, where we look for specific traits to express in plants. This is a far shot question, but how do you think that's gonna sort of have an effect on us, on our genome, maybe?
Oh, that's a
bit philosophical.
Very good question. So the scale that we're looking at here for humans to adapt to a new environment, we are talking like hundreds, if not thousands of generations to actually see changes in the genome. But this all depends on the intensity of the selection that you're looking at. I would say that any innovation, genetic innovation, it's usually much simpler to have it already existing and standing in another species. So precision agriculture, in a sense, we are looking to find innovation that exists or that we can generate to make the plant more resilient. And in terms of food security, I would say that you would never beat diversity. So at the end of the day, diversity is providing the resilience. Diversity is providing with the innovation that you might need.
So you mean diversity in the crops, in the supply chains, and like what we plant?
Yeah, in what we plant, like this is where the robustness stems from. So we might want to go towards introducing more of this diversity to actually face the different challenges in terms of changes.
Yeah, because I mean, from my crude understanding of how agriculture has sort of evolved over the years, it's like now we're at the point where most of the seeds are very similar. For like we have one sort of seed for maize and one for wheat and so on. But you're pointing to a future where we might have so many, like more variants of the same crop.
I wish, definitely. I think this is where you have the highest likelihood of actually having a standing population that can adapt. And I think that's definitely the future might stand in having more of this diversity while keeping standards for production as high as we have them now.
Is this also in a way sort of accelerating also the natural process of plants adapting to different climates? I guess you're sort of making sure that a crop that was maybe originated in latitudes closer to the equator can now also thrive in farther regions.
Yeah, exactly. And this is also how you see that DNA and adaptation, all of this is very dynamic. So this is definitely one goal, to make some species more resistant to climate change, for example. But I think that where precision agriculture really has potential is that you might be changing specifically the genes that you know to be in relationship with this adaptation, but not changing all of the other parts of the DNA that contain the historical diversity that has been accumulating in the population to actually make it resistant to new changes. Let's say that you have if you only have clones and you have a pathogen that's actually very good at infecting this particular clone, you have a catastrophe in terms of losing all of your crops. So precision agriculture in the sense that you could adapt some of the traits, but not all of them, to keep this inherent diversity there.
This is this is really interesting. I heard at some point a while ago that kind of the reason why artificial banana flavor doesn't taste at all like banana is because it is an old cultivar that essentially got wiped out because it was all just clones of each other. And there was this one fungus essentially that infected pretty much the whole population. And so now we use a different cultivar. And so just by having that diversity, it would mean that kind of the production systems are a lot more resilient to various changes that might come down the line.
I didn't know that story. Yeah, that's very funny. And I think that stories like this, you have a lot of them, and some of them with very catastrophic outcomes, like huge famines, for example, because you had like cultivars that were too close to each other, because you have this inbreeding that is allowed in plant species. But yeah, definitely, I think that's one road that we might want to take. And the second one that is also of high importance for human health and precision medicine is how rare variants, so the variants that only you might be carrying, for example, or only a couple of individuals might be carrying, are actually changing the susceptibility to certain diseases. Because you would have it's an additive process by which all of these individual mutations are changing something in terms of your general health, and understanding how they related to the global diseases that we are facing, like chronic illnesses, for example. Can AI actually be used to predict those? Is another it is another like big topic for future developments.
Yeah, of course. I mean, the kind of human health research has massively blossomed with the kind of advent of single nucleotide analysis of positions, etc., etc. So to see the same thing being done in kind of plant genomes is, to me, sounds very natural, but it also sounds super interesting, right? Because again, you have all this diversity now that you're dealing with, and you have very, very serious issues that you're kind of trying to circumvent.
Yeah, definitely. And I think that here the challenge that remains is that most of the diversity in an individual comes from this rare variant, but since they are rare and at low frequency, you do not have a lot of statistical power to actually understand the impact. So you need like other ways to inform you about how this particular change in one letter in one of the 3 billion letters in your human genome is actually going to have like huge impact on your phenotype, like what you can observe.
So are you saying you can use these models to sort of study these rare events much better?
Yeah. In a nutshell, that's okay. What we are doing
because if you, as you said, if you only accumulate data, you would only have like really low frequencies for these events. But I guess with a model like NTV3 or so, you can actually prompt the model to generate these rare variants and then study them, I assume.
Yeah, and also because from the extensive learning that you do on this 9 trillion nucleotide, 150,000 genomes, and all of these omics tracks, you actually put the pieces together of a puzzle where you understand much better not only what the sequence is doing, but also how do all the different tasks relate to each other. And because the model is used to seeing some kind of distribution, whenever you have one of these rare events that is going out of distribution, it can now understand and interpret a little bit more, a little bit better, how is this going to change the overall dynamic of the sequence.
And maybe to wrap this up, so on a personal level, I guess you're working very close to applications, but it still feels a lot like research. You seem very comfortable in this field. How would you say does it compare to your previous work on, like, during your PhD or what you've done before? Do you really like working on applications, or what drives you?
Yeah, I think I really like this new concept. So being able to see DNA, but in through the AI lens and through like a more probabilistic way of understanding what's happening in the genome is really what drives me. I think I'm very curious about what would be the next development. So AI is not my first expertise, but I think it's matches very well the complexity of the genome, and we have this unprecedented opportunity to actually have this non-linear, very deep, complex model to finally match with the complexity of the genomics data itself. So and it helps towards like driving the best applicative cases and applications, because you get to understand the data and to understand how is this going to help your case.
So we've talked a lot about kind of what the strengths of these models are and how this kind of probabilistic approach to looking at genomics is really, really fruitful. And in terms of the kind of NT family of models and where you see their applications, what are the things that you're most excited about? Where would you like to see this work go?
Yeah, I think that where this work is actually already going is extremely exciting, which is the generative biology. So instead of having this model to predict from the DNA sequence, what we are asking them now to do is to synthesize the DNA sequence that is going to match the outcome that is needed. So in a way, you can now prompt the model to actually give you a sequence that is going to lead to a different specificity in terms of tissue, for example, but also in terms of how much of how intense you want the expression to be, for example.
Really, really interesting. Thank you so much for coming on the podcast today and talking to us about all the really interesting work that you're doing. Thank you, Marie.
And thank you both. Yeah, I had a great time.
And to any listeners who might be interested in looking a little bit more at the DeepChain platform and checking out the capabilities thereof, we'll link that in the episode description below.
And make sure to pop by next month, where we will be sticking around the theme of applications, but this time focusing on atomistic simulations and how we're bringing machine learning models into the mix. So see you then.
Bye everyone.